Let me now begin talking about challenging DNA evidence. So there was a report from the National Academy of Sciences that came out exactly 6 years ago called Strengthening Forensic Science. Have you seen that report? It's online and it's free, and it talks about potential issues with forensic science and policy recommendations. And one of the comments they make is about DNA, and what they say, and I'll read this, is that among existing forensic methods, only nuclear DNA analysis has been rigorously shown to have the capacity to consistently and with a high degree of certainty demonstrate a connection between an evidentiary sample and a specific individual or source. And so that's all true, but then there's this little caveat at the bottom that reads, however, there may be problems with how the DNA was interpreted, such as when there are mixed samples. So what I'll be talking about for the next hour or so is what those problems are, the challenges that they pose, both in the data and particularly in the interpretation of mixtures. So just as a review, for people who may not have seen this too often, every human body has trillions of cells, and most of them in each cell there's a nucleus present, and that nucleus contains 2 complete copies of the human genome, which is text that's 3 billion letters long, written in a 4-letter alphabet of the 4 DNA bases, A, C, G, and T. And if we take a look in the middle of the slide at a chromosome, and we were to unravel that chromosome, you'd see the text. The DNA molecule is a linear molecule. It's like a hard drive or a book, for those of you who remember what books are. And there are letters, and those letters spell out either genes, functional proteins, but they're coding instructions for the operating system of the human cell and the human body. And they also contain other sections. In fact, most of the sections, we don't know what they do, and they look like junk DNA or random DNA. One such section is the short tandem repeat. And these are tandemly repeated words that are repeated 10 times, 50 times, and they're scattered throughout the human genome, and there are maybe 100,000 of them, of these paragraphs. And if on the left I'm showing a metaphor of an encyclopedia, there are 23 volumes representing the 23 chromosome pairs that each cell has, one from each parent. If we would take a look at one paragraph, that's a short tandem repeat, this is what it would look like. It has a fixed starting point, shown in red as "take me out to the ball game." It has a fixed ending phrase in the paragraph, say "at the old ball game." I'm using a famous poem. And then in between, there's a fixed region, like "take me out with the crowd, buy me some peanuts and Cracker Jacks," and so on. And then in between these fixed regions of the beginning and the end, there's a word that's repeated. In forensics, it's usually 4 letters from the DNA alphabet. Here, since we already get started with this well-known poem with root, 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 imagine that root's repeated 10 times. If root's repeated 10 times, then that's going to determine the whole length of this paragraph. And that's what DNA testing is about. It's measuring lengths of DNA paragraphs. And in the human population, this particular paragraph was chosen from all the other 100,000 paragraphs because there's a lot of variability at that location. The word root might be repeated 5 times or 6 times or 17 times or 22 times. Typically, there are 15 or 20 different length variants, and now there's a way of distinguishing between people depending on how many times that word is repeated. Different people will have different lengths. So if the word is repeated 10 times, as I indicate on the bottom, then the variant or the allele length is 10 repeats. And so once it's there 10 times, the allele of this paragraph can be described by the number 10, how many times the word root is repeated. What scientists do is they take DNA molecules like a Lexis search. They'll put in "take me out to the ball game" and "at the old ball game," metaphorically. It will go into this encyclopedia and find that paragraph, because there's only one paragraph that reads that way across all 23 volumes. And then they'll look at it, amplify it, make millions of copies of it, and depending on the length of the paragraph, they can then figure out what the allele is, how many times that word was repeated. If the paragraph is shorter, root was repeated fewer times. If the paragraph's longer, root was repeated more times. That's the basis of DNA testing, doing maybe 15 or 20 different tests at the same time to reach into the genetic encyclopedia and find out what these alleles are by measuring lengths of paragraphs to figure out how many words there are, what the allele is. Now, people don't just have one allele, we have two parents, and so our genotypes, which is probably the main concept in all of forensic testing. What is a person's genotype? What's the genotype in the evidence? It's a pair of these alleles. So if you see on the left, there's a chromosome, it's unraveled into a locus. There are two copies, one from the mother, one from the person's father. Maybe at the first repeat in that paragraph in the center, there's 10 repeats of the four-letter word. Then underneath, there's 12 repeats. That person's alleles are a 10 and a 12, and their genotype at that specific location on that chromosome for this one paragraph that's being tested, the short tandem repeat, or STR, is a 10-12. And because there can be 15 or 20 different alleles, there are maybe 100 pairs of alleles. And that's a good way of distinguishing people. The chance that a random person, by coincidence, has the same pair of alleles at that locus, the same genotype as somebody else, is pretty low. And if you test a dozen loci, multiple locations, the chances become maybe 1 in a quadrillion or so, or even less. Okay, there are many, many genotypes, which are like barcodes for people, and there are few people, so genotypes are a way of distinguishing between people. Maybe there are a trillion genotypes, but there's only about 10 billion people on the planet. So the chance that two people have the same genotype is pretty rare. So let's take a look at the simple DNA evidence that scientists saw and testified about before mixtures became prevalent. So imagine there's, on the left, we see DNA from one person. The laboratory takes this DNA and they focus on 10 or 15 different locations, different paragraphs that are short tandem repeats. They amplify and make millions of copies that can be detected with a laser and fluorescence. And for each of these locations, they measure the length of the DNA fragments that were found. So they're measuring the lengths of the paragraphs. And then they use calibrations to figure out how many repeated words there were, how many alleles there are. And in the data that we see in the center, in a simple signal, there would be two peaks. The horizontal axis is increasing length, 10 alleles, 11 alleles, 12 alleles, how many times the word's repeated. In theory, there could be a lot more on the left or a lot more on the right, depending on the lengths of these paragraphs. And then the heights of the peaks correspond to the quantity of DNA. And with simple DNA evidence from the back of the room, you can see there's a peak at 10, there's a peak at 12. Out of the 100 possible allele pairs a person might have had, you can just look at it and see that the answer is a 10 and a 12. That's their genotype. So that's the basis for a lot of what happens in DNA, is that with single source DNA, it's really easy to see what the genotypes are. And there would be, say, 15 of these different experiments. The scientist would look at that data and say, well, at this locus, it's a 10, 12, and they'd write down 15 pairs of numbers that would be a person's genotype. Now, when a comparison is made between the evidence genotype and a known genotype, usually from another person or a suspect, a victim, it could be other evidence, then when that comparison is made and there's a match, the match statistic is depending on how rare that genotype is. It's 1 over the chance of a coincidence. If the chance of a coincidence is 10%, then the match statistic is 1 over 10% or 10. If the rarer it is, the more information is present. So that's the basics of DNA analysis. But now mixtures started appearing 10, 15 years ago, almost as soon as people were doing DNA testing, but with more sensitive kits that have the chemistry, with more sensitive detection with the laser instruments. It became clear that much, if not most, of the evidence were mixtures of 2 or more people. And what happens with a mixture is that one person's DNA, here an eye of a newt, goes into the mix. Another person, maybe an eye of a frog, goes into this cauldron. And It could be 3, 4, 5, or 6 people, but by starting to double the number of people, then you are doubling the amount of work, the toil or trouble that goes into the interpretation and producing a match statistic. Now, the process is the same, but what we're trying to get out to interpret starts getting more interesting. Imagine on the left that there's now 2 people's DNA that are present in the DNA sample, in the swab from a surface or a body cavity. The lab extracts the DNA, amplifies it, separates it out, and at each of these 15 locations develops a signal of low peaks and high peaks that reflect the genotypes that are present. And then the task, and particularly what I'll be talking about today, is how to interpret this mixture evidence by separating the mixture, right? So the mixture was put together and mixed together. Now the task is to separate out what's in that cauldron back into the newt and the frog, to separate out the genotypes from the data for each of the contributors who put their DNA into that sample. Think of it as maybe a victim and a perpetrator, is often what you're seeing in a homicide or a sexual assault. We've previously seen that maybe there's a person with a 10 and a 12. That would be one of the evidence genotypes. And now, looking at the peaks, a person or a computer could say, well, I have a 10, 11, and a 12. The 12 is taller than the 10 and 11. Taller means there's probably more of that allele present, more of that type that's present. But it's usually the case that you can't perfectly separate out what the genotypes are. Because in science, or even weather in Pittsburgh this year, we don't have exact certainty as to what's going to happen, despite all the data that we may have. So maybe the first individual, it could be clear that there's a 10 and a 12, but of the 100 or so allele pair possibilities, what we hope for in science is that the data reduces those possibilities. If when we're done, the possibilities are the same, there's no information. But if we've gone from 100 possibilities, in this example, say, down to 3 possibilities, that reduction of possibility is what's generating the information. And those possibilities are generated with probabilities that go with them, numbers that add up to 100% or 1. And that's what an inferred genotype looks like, that can be derived directly from evidence data corresponding to the DNA that's in the sample, done objectively without looking at the suspect, without looking at the person you're comparing with. Those genotypes can be separated out into a first contributor's genotype, shown in brown, a second contributor's genotype, maybe with some uncertainty. Maybe 10-11 is a possibility, 11-11, 11-12 with different probabilities. Then a comparison can get made to that second contributor's genotype. And when that comparison is made, there can be a match to some extent, but now it's a match to a genotype that's not a definite genotype. It has some probability, it has some uncertainty. And so that 10-12, when it's compared with 10-12, is only there at 50%, because that's the probability of a 10-12. And the result is the match statistic isn't 1 over how rare the genotype is. That match statistic is reduced by the uncertainty. So if there's 50% of the genotype probability present, then that match statistic would become half of what it would have been before. And that's how mixture statistics can be done, by separating out genotypes. And the nice thing about doing it this way is that the math, the concepts, how you explain it to a jury or to a layperson is exactly what we had with single source DNA. If you separate out a mixture into each of the contributors, then by comparing each of the contributors' genotypes with a reference, it's back to comparing one genotype to one genotype, and it becomes simple again.